Hello and welcome yet again to Effortless Mastery. Today we'll be continuing our series on general embryology. And if you are yet to watch the first video, please go back and see the introduction. But today we'll be focusing on cell division. So the big question is, why cell division? Why do I want to know about the cell? How the cell divides? The truth is that every single human being, which is full-grown, multicellular, having up to 10 million cells, 10 raised to the power 14 cells, actually began as one cell, which is called a zygote. So how did I move from one to so many? Simple cell division. There are two types of cell division that we are taught, and those two types of cell division is mitosis and meiosis. Now, for today's class, we'll be learning on mitosis. So our focus will be mitosis. Now, what is mitosis? Mitosis is a type of cell division which involves the formation of a daughter cell or an offspring that has the exact same number of chromosomes as the original. So it is identical. So I'm going to have one cell, which was the parent cell in this case, which was 2n diploid number, dividing into 2. And these two daughter cells are still having diploid, and they are identical to each other. So please observe what I said. Mitosis type of cell division, the parent cell divides into two identical daughter cells that have the exact same number of chromosomes as the parent cell. So there's maintenance of diploid number. What's the key reason? Why mitosis? What's the key reason for mitosis? Mitosis is for two things: growth. The fact is that from small, even as in our first video we made mention of an infant or a neonate, how the neonate moves from the small boy we used to know to a bigger man is by growth. And that growth is actually achieved by mitosis. So the cells keep dividing and dividing. Now, beyond that, another key function of mitosis is tissue repair. Constantly, cells in our body are dying and they ought to be replaced. The replacement of the dead or worn out cell is also achieved by the help of what? Mitosis. It is important to note also critically that mitosis occurs only in somatic cells. What are somatic cells? Somatic cells are the regular body cells other than germ cells. So germ cells are the body cells that will be involved in procreation, that's formation of an offspring. And for humans, the germ cell we have is, or the key germ cell or mature germ cell is sperm and egg. So any other body cell other than sperm and egg is actually going to be called a somatic cell. And that body cell can undergo mitosis. So now mitosis is divided into different phases. Broadly, we look at mitosis first as interphase and then 4 other steps with an acronym PMAT. So first we have an interphase, which some people call pre-mitotic phase. So it is not really part of mitosis, but it is needed. So it's more like a preparatory stage for mitosis, and it is called interphase. After interphase, the main mitotic division starts with P, which is prophase, M, which is metaphase, A, which is anaphase, and T, which is what? Telophase. So after I'm done with this, we now have cytokinesis. That's why some people do /C here. The cell cytoplasm will now divide and we form back a new cell. If it wants to divide again, the new cell has to enter interphase. From interphase, come to prophase, metaphase, anaphase, and telophase. So let's dive right into the details of mitosis. Now, first, under interphase, interphase is divided into 3 steps. Interphase is divided into 3 steps. The 3 steps are G1 phase, S phase, and G2 phase. Now, G1 phase is a period of growth. So before a cell should divide, the cell itself most likely was also formed from a previous division. So after my previous telophase, which was mitotic division, once I am done with the division, first in G1 phase, all I do here, the hallmark is that the cell grows. So most times it is growth in size. Now after G1, in S phase, the key thing that happens that you must always remember is DNA replication. So probably I should bring that and write it out here. So during G1 phase, what's the key thing to note here? Growth. During GS phase, what's the key thing to note here? DNA replication. And I go to the last one, which is G2. During G2 is more like, we say just a period of double checking and maturation. So what happens? In G1, I grow. In ACE, the DNA is replicated. That means the genetic material is duplicated. Now, in G2, I just check back, was there any mistake? If there was a mistake, that cell should not go on, or I correct the mistake. So you double check. Any mistake in replication is corrected in G2. The cell matures and is ready for the main mitosis. So the first step of mitosis is called prophase. Now for prophase, what happens in prophase? So let's go back to interphase. What do we see in the cell? We see a nucleus. We see, this is called a centrosome. A centrosome. And usually a centrosome has two centroles. It's made of two centroles perpendicularly aligned together. So you have a centrole this way, centrole this way. That's what we call a centrosome. Centrosome. Now, so we have, every cell has one centrosome. In fact, centrosome is a characteristic a cell must have for us to know it can do division. So it has a centrosome, which is 2, and then 2 centroles there. Then it has a nucleus, and within it we see some genetic material, which are not quite obvious. So that is what happens in interphase. So once I come to prophase, prophase is, the key things in prophase, number one primary thing is that the chromosomes we're seeing here that were actually appearing thin are not very visible, which we call chromatins, are now quite visible. And because they are visible, that process is called condensation. So under prophase, the key thing under prophase, which must be remembered, is that number one thing that happens in prophase is condensation of chromosomes. Now, aside from condensation, another thing that happens in prophase, which we see, is that the centrosome, which was initially 1, remember I told you centrosome is 2 centroles. The centrosome, which was initially 1, duplicates itself, and we now have 2 centrosomes. And these 2 centrosomes actually migrate to different poles. So we have duplication and migration of centrosome. So please observe. So within here, in prophase, simple thing that happens, number 1, condensation, C, alphabet C, condensation of what? Centrosome. So chromosomes. Initially, the chromosomes were so thin, not very visible, but now they condense, so they become visible. In the second thing that happens here is duplication. So the centrosome that was initially 1 now becomes 2, and they migrate to different poles. The third thing that occurs is that the nuclear membrane disintegrates. So I use an acronym for this, and it is CDD. So the hallmark of everything that happens in prophase is 3 things. Number 1, condensation of chromatin into chromosomes that are actually visible. You see, why is it visible? You have seen 2 sister chromatids joined together by a centromere. Joined together by a centromere. Then you now see duplication of who? The centrose that was initially 1 is now 2. And then number 3 is now that I have disintegration of the nuclear membrane. Some people make mention of a 4th thing, which is formation of mitotic spindle. And they say it happens in late prophase. Other people believe that mitotic spindle actually form in another phase called pro-metaphase. But whatsoever you are handed to, it is the same thing. So for the sake of today's class, I gave you 4 steps. So I will add it here and say formation of a mitotic spindle. So what are the 4 things we have in prophase? C. C is for what? Alphabet C, condensation. Alphabet D, duplication and migration. The next D, disintegration of nuclear membrane. The last F is formation of mitotic spindle. So they start forming. Now in metaphase, the key thing in metaphase you must always remember is alignment. Hear the word alignment and then hear the word equatorial plane. So all of the 3 chromosomes I had here are now aligned in the middle. So the mitotic spindles attach firmly now. So that spindles are just microtubules that arise from one centrosome to attach to the to the chromosome in the middle. So you realize from here, I have these people arising and heading here, arising and heading here. Now, the exact point, remember I said every chromosome has two sister chromatids joined together by a centromere. Now, the exact point that mitotic spindles are attached to is called kinetochore. Kinetochore. So this kinetochore is actually, it's just a protein on the centrosome. So on the centromere, So that's where they attach, kinetochore. All right, so what happens in metaphase? These guys are lying in the midline called equatorial plane. And then we now go to the next step, which is anaphase. What happens in anaphase? Simple. In anaphase, they split. So the word for anaphase here will be split. So the chromosome will split at the centromere such that I have sister chromatids now appearing as single chromosomes to either sides. And the key thing about the division here is that the division, for me to form identical, realize that every single person is cutting into two. And we come to telophase. Telophase is the exact opposite of prophase. What happened in prophase? Condensation of chromosome. Here, what will happen in telophase? What was visible becomes diffused. So it's no more condensed, but rather it is diffused. And then I had duplication of centrosome. Here I'm just having one centrosome at either end. The nuclear membrane that disintegrated is now reformed. Mitotic spindle that was formed is no more found here. So those are the four things. A direct opposite of these four happens here. And after telophase, the cytoplasm now divides into two to form two new cells. And then we are at the end of mitosis. So in summary, what have we learned about mitosis today? Mitosis is a type of cell division whereby a single parent cell divides into two identical daughter cells. And these two daughter cells are similar in their number of chromosomes as the parents. So there is maintenance of diploid number of chromosomes, which is 2n. Now, where does mitosis occur? It occurs in cells called somatic cells. What is the purpose of mitosis? Growth and tissue repair. Now, the stages of mitosis are broadly divided into pre-mitotic, which is called interphase, and the main mitotic phase. The pre-mitotic, which is interphase, is that I have a cell just returning from a previous mitosis, undergoing first G1 phase, which is growth, S phase, which is DNA replication, and then G2 phase, which is double checking and maturation. Following that, there is prophase, where four key things happen. Condensation of what? My chromosomes, which were initially thin-like, to be more visible as two sister chromatids joined together by a centromere. Beyond that, we have items we call centrosome, which actually duplicate and migrate to opposite pole. Then the next thing is that the nuclear membrane I usually had now disintegrates so that I no more have a nuclear membrane. And finally, mitotic spindles begin to form, which are just microtubules. Now, in metaphase, these people align. The chromosomes now align in the midline called the equatorial plane. But something is important. I said the exact point of the centromere that the mitotic spindle will attach to. So when they come, they attach to an exact point, and that is called kinetochore. It's a protein, a kinetochore. From here, in anaphase, there's splitting, whereby the sister chromatids are split from each other at the centromere. And then in telophase, the reversal of all of this happen. So thank you very much for joining us. I hope you clearly understood mitosis. So in our next class, we'll focus on meiosis. So please do well to subscribe to this YouTube channel, like this video, and then comment in the section below. And this will help push the video to many others.